Okay, so hello and welcome back to Agriculture Academy. So today we'll be talking about, we spoke about the ER, how the resident proteins are there, how they help in folding of the proteins, right? So we have discussed about the BIP proteins and the PDI. Now about the quality control in ER. So what does it mean? So the many proteins which are synthesized in the ER rapidly get degraded because they fail to fold properly. Okay, now suppose these misfolded proteins are there, so we have already said that they are going for the degradation. So this process is known as ER associated degradation. Now, in which the misfolded proteins are identified, they returned from the ER to the cytosol, because in the cytosol only you have got your ubiquitin proteasome system, where the proteins are getting degraded and all the amino acids are free into the cytosol. Now, because they assist proteins in correct folding, chaperones and protein processing enzymes in the ER lumen often act as sensors of the misfolded proteins. So, this chaperones, for example, like in the previous lecture, we talked about it, right? So, BIP proteins are actually nothing but your chaperones. So, they help in the proper folding of the proteins. Now, what chaperones does? They actually help in forming of the covalent bond or breaking of the covalent bond, so that the correct protein is getting folded. Yeah, so these chaperones, they form the covalent bond or they break the covalent bond so that the proteins are actually getting properly folded. Now, so these chaperones, they are responsible for the folding of the protein, so they will act as the sensor also. If suppose the proteins are not getting properly folded, so they'll understand, okay, this is misfolded, this cannot be rectified, so they should go for the ERAD pathway. What is ERAD? ER associated degradation. Okay, please do remember this thing. Now, one well-characterized pathway of glycoprotein folding involves chaperones that recognize the partially processed oligosaccharides from which terminal glucose residues have been removed. So if you remember, in the previous class, we talked about it, right? The dolicol, of which 2 NAG, 9 mannose, and 3 glucose were there, if you remember. And I said, once it starts getting the processing of the protein, the glucose are removed. Okay, so it is being understood by those chaperones, and then they help in the glycoprotein folding so that it goes into the correct formation. Now, if the glycoprotein is correctly folded, it will go to the Golgi for further modification. Okay, now we need to talk about these proteins also, like how it is helping. So in the case of the glycoprotein folding, as you see, the ribosomes are there, translocations, so your protein is present over here. Then from the dolicol, sorry, from the dolicol, the oligosaccharide has been transferred. Okay, the 14 sugar oligosaccharide is being transferred to your protein. Now, this chaperone, can you see how beautifully it is actually making an arrangements or making interactions with the protein as well as the sugar moiety over here? Here. So it understands that already 3 glucose were removed. So it will help in the proper folding of the protein. So it gets correctly folded. Now in the vesicle, it gets loaded and goes to the Golgi apparatus. Okay. Now, if it understands that the glucose are not removed, or it is, not properly folded, it gets misfolded, then they are gone for the ubiquitin ligase complex where the degradation is happening. Okay. Clear? This path is clear? Okay. So, however, if the glycoprotein fails to fold, it is instead targeted to the ERAD pathway, and the misfolded protein is transferred to the transmembrane complex with the ubiquitin ligase, translocated back to the cytosol where it gets degraded in the proteasome. So, this already we have discussed, like how proteasome is actually helping in the degradation. So, E1, E2, E3 is there. E1 carries a ubiquitin on ATP activation. This ubiquitin molecule is transferred to the E2, and E3 will help in transferring this ubiquitin molecule to the protein from E2. So this is how it keeps on adding the ubiquitin molecule to the protein, and finally it is taken into the proteasome, where in the proteasome ubiquitin molecules and the rest of the proteins are degraded into the peptides or small amino acids. Clear? This is just a repetition of the same slide. Now, the level of unfolded proteins in the ER is monitored in order to coordinate the protein folding capacity of the ER with the physiological needs of the cell. Sometimes, see, what happens is that if suppose your cell is not getting the nourishment, okay, it means it is not getting the new resources, new amino acids or new DNTPs. If they are not getting it, sometimes what the cell does, it is gonna make or it is going to make few of your proteins to undergo degradation so that they can reuse the amino acid cycle, amino acids for the new synthesis of the proteins, which is that time required. Okay, so this also happens inside the cell when the new resources are not being fulfilled. Okay, now the level of unfolded proteins in the ER is monitored in order to coordinate the protein folding capacity of the ER with the physiological needs of the cell. This regulation is mediated by the signaling pathway known as UPR, unfolded protein response, which is activated if an excess of unfolded proteins accumulates in the ER. See, if suppose the chaperones have done their job and you have got unwanted expression of the protein, okay, unwanted expression of the protein, and it is kept unfolded only. Okay, the chaperones have not converted them into a folded proteins or functional proteins. So this will lead, first thing, either you will need more protection of the chaperones or they can help it, or you need to remove the unfolded proteins. Okay, that is to stop the synthesis of the new proteins entering. Did you get the point over here, or I just made it more confusing? Did you get the point here? See, unfolded proteins दो तरीके से रहेंगे आपके सिस्टम में. Either the chaperones are not able to fold, or it is high in expression, more than required. ठीक है, तो दो रिस्पॉन्स क्या होंगे कि आप या फिर chaperone का एक्सप्रेशन बढ़ाओ, या अनफोल्डेड जो, और या फिर जो न्यूली सिंथेसाइज्ड प्रोटीन्स है that needs to be removed. Did you get the point over here, Krishita?
Yes, ma'am.
Okay. Now, so how this UPR is actually responding. So we have got the unfolded proteins which activate 3 receptors in the ER membrane. Okay. First one is your IRE, which will cleave your pre-mRNA for the transcription factor XPP1, which will lead to the synthesis of XPP1, which translocates to the nucleus and stimulates transcription of the UPR target genes. ठीक है, तो सबसे पहले आपका response क्या था? IRE1 का. Okay. Now, so IRE1, what it is going to do? It is going to lead, or it is going to cleave the pre-mRNA of your transcription factor, which leads to the synthesis of this. Okay, so it will go to the nucleus. Here you can see you have got the IRE1. It goes for the cleavage of your mRNA. Now this mRNA results in the transcription factor. This transcription factor goes inside your nucleus, where the UPR targeted genes are being activated. Second क्या हो गया आपका ATF6, which is cleave to release the active form of the transcription factor. Okay, so यहाँ पे ATF6 है, it gets activated, it is going to cleave and the transcription factor gets activated, reaches the nucleus. The third one is PERK. It's a protein kinase that phosphorylates translation factor EIF2, eukaryotic initiation factor 2, if you remember from the translation. Okay, now this inhibits general translation, reducing the amount of protein entering the ER. So here you can see how these receptors are getting activated, because they see that there are so much expression of the unfolded proteins are present. Okay, so when they interact here, these interactions, okay, this results in the activation of your IRE1, ATF6 and PERK, eventually resulting in the transcription factor activation, which goes into the nucleus, and nucleus, the gene expression is for the UPR targeted genes and to stop the translation. Okay, it also it also results in preferential translation of the transcription factor ATF4, which further contributes to the induction of your UPR target genes encoding chaperones or the enzymes involved in lipid synthesis and ERAD proteins. ERAD क्या था आपका? ER associated degradation. ठीक है, so this is how the unfolded protein response is being regulated. एक क्या हो गया, या फिर आप उसका ट्रांसलेशन बंद करोगे, या फिर आप उसका chaperone से उसका फोल्डिंग इनिशिएट करोगे, और यू आर गोइंग टू target these proteins so that it can go for the degradation. So this is how you are going to maintain the level of the unfolded protein. Did you guys get this point? Take your time, go through the figure, and let me know like who all can explain this thing. Yes. Akshay, would you like to try explaining this UPR?
Okay, ma'am. So if there is a lot of unfolded proteins, it means that there is overexpression of unfolded proteins present. So what happens is that these receptors get activated, IRE1, ATF6 and PREK. So when IRE1 gets activated, it goes for pre-mRNA cleavage, and that initiates XP1 mRNA, and then the XBB1 transcription factor, and then it goes into the nucleus, and where it gets folded with the help of chaperones and other enzymes.
No. Transcription factor, it is going to help in the expression of the UPR target genes for like chaperones or ER proteins. Okay, so these transcription factor kya karta hai? It is for the gene expression. Okay, it does not get folded. It is the transcription factor required for the synthesis of your chaperones or ERID associated proteins. Okay, yeah. Then
in the same way, like PREK, when it gets phosphorylated, it inhibits the translation, where and this also, so when it gets inhibits the translation, this ATF4 goes and binds, and again it helps in encoding the genes for again chaperones, lipid synthesis and enzymes. Again, in the same way, like ATF6, it undergoes cleavage, and then again it goes and encodes the target genes and the chaperones needed for the folding of proteins.
okay, so here what happens in the case of PERK is that it's a kinase. Okay, so kinase adds phosphate group to your initiation factor. This initiation factor is for your translation. तो इसके वजह से क्या होगा, ट्रांसलेशन आपका इनहिबिट हो जाएगा, which is happening in the cytosol, right? Now this inhibition, okay, it is gonna make sure that this ATF4 is also getting activated. Okay, this is a transcription factor which will go and help in this expression of the gene. Okay, it is not that the PERK is doing something. PERK is a kinase enzyme which inhibits which phosphorylates initiation factor of the translation, hence inhibiting the translation in the cytosol, which eventually results in the activation of your ATF4, which goes into the nucleus. Similarly, here ATF6 is there, it's a receptor, right? Now, when it goes and binds to your unfolded proteins, high expression, then this gets cleaved from here. And this particular domain goes as your transcription factor into the nucleus. Now, little more clear?
Yes, ma'am.
Okay. So if these changes are insufficient to adjust protein folding in the ER to a normal level, sustained activity of the unfolded protein response leads to programmed cell death, thereby eliminating the cells that are unable to properly fold proteins from the body. See, even though we have got so much of mechanism, like just now we see that how unfolded protein is there, and if suppose these regulation is not helping in bringing down to the normal level, then the entire cell gets a signal that it is going to be dangerous. Okay, what happens is that when you have got more expression, overexpression of a certain protein, it might result in the dysregulation of your cell cycle activity, which eventually results in the cancer onset. Okay, So instead of a cell becoming a cancer in nature, what it understands is that instead of becoming a cancer, it's better to undergo apoptosis. Okay, so it will understand that, okay, my system fails in regulating the unfolded protein response, so which will result in the bad activity of the cell. So it is going to undergo programmed cell death, and here what it means is the apoptosis. Okay. Okay, so about the chaperones and the chaperonins. So I thought of discussing this like yesterday you have asked. Did you guys get the answer? Did you try searching like the difference?
So I have a doubt. There is this program cell death that you spoke about. For that, all the three pathways should be functional, or even if one doesn't work, still the cell would enter into the program cell death.
No. See, it again depends on the level of the UPR response. See, if you have got very high unfolded proteins, then definitely all the three pathways should be activated. Okay. If your translation is not happening, then I don't want third pathway to be activated, right? Simply inhibiting the translation. So only first two pathways are enough. Eventually, what is that? They all work in the synergistic. Right now, I don't have any data saying that only one pathway is activated or two or all the three. What I understand from the reference book is that these three should work in synergistic way so that this unfolded protein should go back to the normal state. If it is not going just because of one of the pathways, then it might go for the program cell death.
Okay.
Huh? Okay. Okay, what about this chaperones and chaperonins? Did anyone try to understand the difference? So here the example we have talked about HSP70, heat shock proteins. Okay, so here see chaperones, as we just saw in the previous lecture, they bind to your polypeptide chain like this. It was shown, right? So they help in the covalent bond formation, okay, either breaking or making, so that the proteins are getting properly folded. Okay, now coming to the chaperonins. Chaperonins are, they provide environment wherein which the protein gets folded. For example, they are like a big hollow kind of protein subunit into which the protein goes inside. Okay, यहाँ पे क्या होगा, जैसे ये प्रोटीन है, ये HSP है, okay, some HSP90 is there. So these HSP90 make sure that the unfolded proteins remains like this in the unfolded form. So that will be taken inside of this chaperonin, where HSP90 आपके फिर बाहर रह जाएंगे and only polypeptide chain will go. Now this will go under certain modifications which eventually results in the proper folding of the protein. So it provides an environment inside which the single polypeptide chain gets folded properly. यहाँ पे आप misfolded protein भी ला सकते हो with the help of the HSP90. This goes inside and gets properly folded. इतना करने के बावजूद, if these proteins are not getting folded, then they go for the degradation. Now the difference is clear. So from the figure, if you see, you can say that chaperones are small proteins. Chaperonins are huge, big protein subunit domains are there. Okay, example HSP70 हो गया, chaperonins का grow EL and grow ES करके है. We'll come to that. So this is like, you know, a container, फिर उसको एक lid रहता है. So the moment protein enters, the lid gets closed. It makes sure the protein is getting folded and then comes out. Did you get the difference now? So both are responsible for proper folding of the proteins.
Yes, ma'am. Ma'am, also I read somewhere regarding chaperones that chaperones are not involved in folding of the proteins, rather they prevent improper folding. So is that correct? Because I read it.
Yes. So HSP70 will help. See, that's why I was saying covalent bond breaking and making. So HSP70 will prevent misfolding of the proteins, but we talked about BIP, right? So BIP is responsible for the folding of the proteins.
Okay.
हाँ, HSP 70, 90 ये क्या हो गए जो आपका प्रिवेंट करेगा मिसफोल्डिंग, where BIP is helping in the folding of the protein. So we have got so many chaperones and few of the chaperonins. Okay, let's move ahead then. Now coming to the smooth ER and lipid synthesis. So we know SER or smooth endoplasmic reticulum is responsible for your lipid synthesis. Okay. So because they're extremely, in addition to its activities in the processing of secreted and membrane proteins, ER is also meant for the lipids. Because they're extremely hydrophobic, membrane lipids are synthesized in association with already existing membrane rather than in the aqueous environment of the cytosol. So some lipids are synthesized in association with the other membranes. Most are synthesized in the ER, and then this ER, this particular part is then destined to the other membranes. यानी कि क्या वो synthesize करके vesicle form करेगा. This vesicle will get fused to the plasma membrane then. For example, phosphatidylcholine is synthesized in the ER and then transferred to the mitochondria, where it is going to get inserted. We'll come to that. With figure, it becomes more clear actually. So the membranes of the eukaryotic cells, they are composed of three main lipids: phospholipids, glycolipids, cholesterol. We all saw that, and they have got the base of the glycerol. Okay. Now they synthesize on the cytosolic side of the ER membrane from water soluble cytosolic precursor. ठीक है, अब इसकी वजह से क्या होगा, cytosolic side में ही होगा. So what is happening then? The cytosolic part of the ER will get increase in the length. Okay, and then the role of the lipase floppase comes. Okay, we'll come to that eventually. Fatty acids are then transferred from coenzyme A carriers to the glycerol 3-phosphate by membrane bound enzymes, and the resulting phospholipid is inserted into the membrane. Okay, so this is how. Don't worry, looking at the structure, you don't have to remember, that's okay. So this is your base. Okay, to this if you add ethanolamine, it becomes your PE, choline it becomes PC, serine means PS, inositol group for PI, and then you have got this sphingomyelin. Okay, so the main base is over here to the glycerol. When you add a phosphate group and 2 fatty acid, it becomes a phosphatidic acid. To this, when you add the certain groups, you get different types of the phospholipids. Okay. So here, the structure of the glycolipids. So when you say glyco, of course, you're going to have a sugar moiety present to that. Okay. Now, cholesterol and steroid hormones, as you know, most of these steroid hormones, the base is the cholesterol. Depending on the functional group, it just changes, and eventually you get testosterone, estradiol, for examples. Now, synthesis of a phospholipid. Let's see how it is being done. So first, glycerol phospholipids are synthesized in the ER membrane from the cytosolic precursor. So always remember this is happening at the cytosolic part. So here you can see that you have got the phosphatidic acid. Okay. Now phosphatidic acid, you got it from your glycerol 3-phosphate. 2 fatty acid chains were attached plus the phosphate group. So here you can see the phosphate group is there. This is your 3-carbon. Okay, this is your 3-carbon glycerol, to which first and second the fatty acid groups are added. Okay, R1 and R2. To the third phosphate group is present. Now, when you remove this phosphate group, okay, it becomes your diacyl glycerol. Then you have added choline group. Choline, when it is removed, the CMP goes and only choline group has been added. So this is how it becomes your PC. Ab choline ki jagah serine aayega, so it becomes your PS. Right? Inositol आएगा तो वो PI हो जाएगा. And this is how the phospholipids are synthesized. ठीक है? Did you get the point over here? अब क्वेश्चन ये आना चाहिए कि ये कैसे बढ़ रहा है. So कैसे रहता है, इन दोनों के बीच में एक phosphatidic acid insert हो जाएगा. And to this you start adding. So बीच में यहाँ पे add हो जाएगा. So what happens to the other side? Plasma membrane के this side, the another side which is towards the lumen side, there will be a gap. Okay? So it looks like this. And you have just imagine, let's say that these are the new ones. ऐसे. So इस साइड क्या होगा, आपको गैप आ जाएगा. Correct? So what happens, then later they get flip. कुछ यहाँ चले जाते हैं, कुछ वहां चले जाते हैं, and then they balance the proper length of the plasma membrane. Okay, the same thing is written what we have just discussed now. Now see, जैसे ही आपका ER membrane है, phospholipid synthesis हो गया, so you can see that this yellow color things are extra which were added, and it is added only to the cytosolic side. So जो आपका lumen side में है, वहाँ पे gap आ जाता है. So now in order to balance it with the help of the flipases, what happens? Few phospholipids will go to the other side. Okay, and that's how the entire length or the growth of the phospholipid bilayer is maintained. So eventually what had happened? You have increased the phospholipid, right? आपका growth हो गया है, and that's how the lipid synthesis happens for most of your membranes. Yes, did you get all this, everyone?
Yes, ma'am.
Okay. Okay, so now the export of proteins and lipids from the ER. As we have discussed in the case of the nucleus, there are certain sequences, right? So that it is understood like what kind of protein should be taken in or what should be exported out, right? In the same way, for ER also, there are specific sequences which you have to remember. Now look at this. So this is the vesicular transport between the ER and Golgi. And do understand that this is like from ER to Golgi it is going. Okay, so what do we see here? The proteins हो गया या lipid हो गया आपका, ये सब आपको सबसे पहले ER से Golgi में जाएगा, then only it goes to its final destination. Okay, so as we know that they actually have got the ER exit sites, वहीं से ही आपका vesicle बनता है. Okay, so let's say that this vesicle is budding, so it is carrying all the export signal. Can you see this? They have highlighted over here, and inside that you have got your proteins. ठीक है, और जो भी lipid हो गया, if it is meant for the distribution. So this vesicle will get exit from the ERES site. Vesicle कहाँ से बनेगा आपका? जहाँ पे ERES site है, that is for your ER exit sites. Okay. So from there it will form and it will get fused to your ERGIC, that is your Golgi ER Golgi intermediate compartment, जो आपके ER और Golgi apparatus के बीच में आता है, ERGIC. It goes, it goes and gets fused over there, ठीक है? And then it comes to the Golgi. Now what happens is that so the proteins which is meant for your Golgi, okay? Which is meant for your ER. As we have discussed that there are resident proteins, for example, BIP हो गया, आपका PDI हो गया, right? So these are meant to be present only in the ER, but sometimes because they are also present in the lumen along with the target proteins, they also get collected in these vesicles, and by mistake from the vesicle, it goes to the Golgi. Okay, so the receptor, there are certain receptors known as KDEL receptor. So they identify that these proteins which have got the KDEL sequences, they are meant to be present in the ER. So the binding of the KDEL receptor with the KDEL will make sure or ensure that these proteins are taken back in the form of the vesicle and gets fused with the ER back. So this is how you make sure that your resident ER proteins remain in the lumen of the ER and they are marked as KDEL. KDEL क्या होगा यहाँ पे आपका? Those are the amino acid sequence. Okay, so they are present at the C terminus. So KDEL, they are the sequences which is present at the C terminus. So if these proteins are exported from the ER to the Golgi, they recognize by the recycling receptor known as KDEL receptor and then selectively return back to the ER. So what is that which we have to remember here? The sequences, it is the KDEL sequences. Okay. Now again, KDEL sequences is meant for your soluble proteins. It is for your soluble proteins. So what is the sequence? KDEL. If it is your membrane bound proteins, then the sequence is KKXX. Got it? So KDEL and KKXX, these are the sequences. It means those proteins are resident to ER. So they should be present there. If by mistake they go to the Golgi, then with the help of the recycling receptor, they should be retrieved back to the ER. Did you get this point? Everyone, Tanvi? Somewhere, anywhere, do I need to repeat?
Ma'am, what was that KKXX that you wrote?
It is a signal for membrane bound proteins which are resident to ER. For soluble, it is Cadal sequences, and KKXX is for your membrane bound proteins, which are supposed to be present on the ER membrane. So vesicle जब form होता है, so vesicle formation means what? इतना part of membrane is going, right? So sometimes along with the other proteins, the ER resident membrane proteins also gets, transported by mistake. So then they recycle back. Okay. Okay, so it's the same thing. Unmarked proteins in the ER can also be packaged into the vesicles and transported to the Golgi by a default pathway, so which does not require specific signals for protein packaging. Now, not only the proteins which is destined for export, but also proteins which are resident to ER, they also get packaged. So if these proteins were allowed to proceed along the secretory pathway, they would be lost to the cell. And that's why this sequences helps in bringing them back. Okay, so the Cadal sequence, which is present at the C terminus, that is your lysine, aspartic acid, glutamine, and leucine. So they are responsible for the retrieval, so that these proteins, the resident proteins, can be brought back to the ER. Okay, so along with the Cadal, we have got the KKXS. So this is for your ER transmembrane proteins. Okay, yeah, that's all. So here, this is the end of the ER. Okay, so all related important topics which was included in the ER as part of CSS syllabus has been completed. So here ER gets completed, and the next topic what we will take in future is the Golgi apparatus. Okay.